Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Hello Anthony, Abhi, a very happy good evening. How are you? Good. So for all those who are new to my session, so Monday, Wednesday and Friday I teach for a class 12 topic, and Tuesday, Thursday, Saturday I teach a class 11 topic. So today we are into the second topic of this unit, reproduction. So we are going to talk about sexual reproduction in flowering plants. So ready? So it's a chapter which is equally important. So this has a weightage of 5%. So shall we go forward? Hello sir. So thank you so much for joining our session today. So please do like, share and subscribe. Okay guys, so please do like, share and subscribe. So let your friends know that we are here talking about the entire syllabus of biology. So let your friends know so that they will watch. Okay? Okay. So before we actually move into today's topic, so I have a few announcements to make. So shall we go and see what announcements that I have? Yes? Okay. So the announcement is, so I have a mentee today at 7 p.m. So the mentee is from the topic biodiversity and conservation. So please do join me at 7 p.m. this evening. Alright? Yes? Okay. So what are the topics that we are going to discuss in this chapter? So once again I am repeating, so we are going to have a mentee at 7 p.m. this evening. So please do join me for the mentee, and it is from biodiversity and conservation. So same 15 questions, 20 seconds each. Alright? So what are the topics that we are going to discuss in this chapter? So number one, so let me tell you, so this chapter has a weightage of 5%. So which means this chapter is important, and there are questions in this topic which can easily confuse you. So pay attention. Alright? Yes. And one small correction. So yesterday at 7 o'clock session where I was giving a recap of cell cycle and cell division. So there was one question which said, pairing of chromosome starts during which of the following phases of meiosis 1? I said packeting. No, the answer is zygote. So the pairing of chromosome begins in zygote, but crossing over happens during packeting. Okay, so kindly make a change of it. Okay. So I assumed it as crossing over, and hence I said it as packeting, but pairing starts at zygote stage. Okay, so please make a note. If you have made notes, so kindly change it there. All right? Okay. So these are the topics that we'll be talking about in this chapter. The first one is I'll tell you why we are studying about this. Then I think you know the worlds of a flower because we were done with morphology of this chapter. Then we'll talk about pre-fertilization events, the structure and the events. So first we'll talk about the male reproductive part of a flower, that is stamens, microsporangium and pollen grains. Then we'll move into pistil, megasporangium and embryo sac. So followed by, so this is pre-fertilization. So pre-fertilization is when your gametes are formed. So for the formation of gametes, you need the male and the female reproductive system, followed by the transfer of the male gamete to the female genital tract. So that is pollination. So once pollination happens, you have fertilization. So here in case of plants, it is double fertilization. Then post-fertilization, what are the changes and events that you're going to have? Sorry. So here we'll talk about the development of embryo, the development of endosperm, C, and finally we have this one topic known as apomixis and polyembryony. So we will see all this. So this is an overview of this chapter. Yes sir, a very happy good evening. So thank you so much for joining our session. So please do like, share and subscribe. Alright, so shall we go and talk about flowers now? So shall we talk about why you should study about this chapter? So this unit is reproduction, right? So in the first topic, reproduction in organisms, I was telling you why reproduction is an important process, right? So that is to ensure the continuity of species from one generation to other generation. So that is why reproduction is needed. So now in the next chapter, we are talking about how flowering plants are going to reproduce. So if the plant does not have a flower, then it is going to reproduce by vegetative means, right? So that is your rhizome, your runner, stolon, etc. So all that we have seen in morphology. Now, if a plant has flowers, then how is it going to be reproduced? So what is the mode of reproduction there? All right. So you completed reproduction in organisms. All right. Very good, Anthony Abhi. Very good. So start making a timetable so that you can finish portions very quickly and start revision. Okay. Fine. Okay. So here we will talk about, so before we go into this chapter, so flowers are always very close to humankind, right? So we use flowers for a lot of purpose, like when we have to express love, we give flowers. At the same time, when you have to mount, you give flowers, right? So flower is used for a combination of things. So flower usually represents mixed feeling in your human world, right? So flowers are of various types, so flowers are colorful. So till now we don't know exactly so many varieties of flowers are there. So still some varieties are yet to be discovered. So like this, a lot of history related with flowers are present. Hello, hi Krishna Kumar. Thank you so much for joining my session. So please do like, share and subscribe. Alright. So from the red roses to various types of roses and various kind of flowers. So you have a lot of things. So you have a myriad of flowers, and flowers always, so one good thing about the flower is a flower does not regret that it's going to die very soon, right? So what do you think is the lifespan of a flower? Hardly 3 to 4 days, right? But a flower never is sad that it's going to die, right? And a flower is never sad that it has a very short lifespan. So always people say, be like a flower. So even so its fragrance, its values only for a small period of time. So you pluck a flower, you keep it with you, but how long will you admire it? As long as it's fresh and beautiful, right? So that is one beauty of the flower and one lesson from the flower that you should take. Alright, so flowers are used for aesthetic, ornamental, social, religious and cultural values. Okay, so I hope you remember what are the parts of a flower, right? So shall we quickly recap the parts of a flower now? Yes? Okay, so talking first about the parts of a flower. So here we go. So what are the 4 whorls of a flower? So we have 4 whorls in a flower, right? So the 4 whorls are present on what? The 4 whorls are situated on a receptacle or on your thalamus, right? And what are the 4 whorls of your flower? So it starts with your calyx. So calyx are your sepals. Then you have your corolla. Corolla are your petals. Right? Then you have your androecium. So androecium is the male part of the plant. Yes. And 4th one is your gynoecium, that is the female portion of the plants. So these are the 4 whorls of a flower. So if a flower has both androecium and gynoecium, it is known as a bisexual flower, meaning it has both the sexes. Okay, and when it has only one, either androecium or gynoecium, it is a unisexual flower. So if a plant has only androecium, it is called as a staminate, meaning it has only the male part. If it has only gynoecium, it is called as pistillate. It has only the female portion. And if a particular flower has all the 4 whorls, it is called as a complete flower. Right? So here is where you have a receptacle. So receptacle is the place or the platform from which all the 4 whorls arises. So here you have your sepals. Okay, so sepals are your calyx. That's what I've written here. Then you have your petals. So petals are the colorful ones. So these are your petals. Okay, they are known as corolla. Then you have your androecium, that is your anther, and your filament, that is your stamen and filament together forms your androecium. Okay, then you have your stigma. So stigma style ovary forms your carpel. Yes, monoecious and dioecious. So which is monoecious? A plant having bisexual flower is monoecious, and a plant having unisexual flower is dioecious. So monoecious plant, only one plant is required to complete the life cycle of the organism. So when I say dioecious plant, so 2 plants, male plant and female plant, are required to complete the reproductive cycle of a plant. Okay. Yes, good Anthony Abhi. Hello Satyendra, hello, hi. So thank you so much for joining me. So please do like, share and subscribe. So shall we go forward? Yes? Alright, so this is done. So now we'll talk about the male part. So pre-fertilization events and structures. So what are they? So there are hormones. So what are the 3 phases in a plant? So in the previous chapter, reproduction in organism, so we were talking about 3 phases, right? So what are they? So first is your birth phase, or you can say the growth phase or the juvenile phase. Then you have the reproductive phase. Then you have the senescent phase, right? So you have 3 stages in your plant, okay? When is the test on reproduction chapter? So maybe next week, okay? So today I have a Menti quiz at 7 p.m. and it is from biodiversity and conservation. From next week I will start the Menti quiz from reproduction, alright? Okay, so anyways, thank you Kumar for joining us. So please do like, share and subscribe. Yes, you can say vegetative phase, juvenile phase and senescent phase. Yes, you're okay. So juvenile phase is in case of animals. So vegetative phase in case of plants. Very good, Antonia B. Okay. So, to begin the reproductive phase of the plant, so you have some hormonal and structural changes which are initiated. Alright, so there is some differentiation and development of your floral primordium because your shoot axis, shoot apical meristem is modified to become into a flower, right? So then you have the development of inflorescence and then a bud arises and then you have a flower coming out. Okay, so the flower can be of two, the flower possesses two parts, one is andrecium, the other one is gynoecium. So andrecium is the male part and gynoecium is the female part. Alright, yes. So this is how it starts. So first you have a bud and then you have a flower. Alright, so till this much it's clear. So now shall we move into the male reproductive part of the flower, that is your stamens. Okay, fine. So here we move. Stamen, microsporangium and pollen grain. So PG in our language stands for your pollen grains. Okay. So here we go. So stamen has two parts. One is your filament, the other one is your anther. So andrecium or stamen, both are the entire male reproductive part is known as andrecium. We call that andrecium and the structure is called as a stamen, and stamen has two parts, filament and anther. So you have a long filament and then the terminally bilobed structure is your anther. Okay, so this is your filament and this is your anther. Fine. So the filament is long. Yes, of course it is long. It is slender stalk and its proximal end is attached to the thalamus or the petal. So this is either attached to the thalamus from which all the four whorls of a flower arises, or it is attached to the petals. When stamens are attached to the petals, can you recollect what is that condition called as? It is called as epipetalis condition, right? So when we were talking about your morphology, I have told you, if stamens are fused with your petals, the condition is called as epipetalis condition. So this is how you keep recollecting the previously learned topic so that you do not miss any concept. Fine? Okay. So the number and length of stamens, so here I have drawn this length, but this is not a fixed length, right? So each flower can have different length and many number of stamens as well. So if stamens are fused together, it is usually in three types: monadelphous, diadelphous and polyadelphous. It is in bunches, right? If it is one bunch, it is monadelphous, two bunch diadelphous, and many bunches, it is polyadelphous, right? But at the same time, if stamens are free, that condition is called as polyandrous condition. So remember that. So this length and the number of stamens vary in each case. Hello Vinu, hi, a very happy good evening. So thank you so much for joining. So how are your school exams going on? Good. So please do like, share and subscribe. So Vinu, I have a Menti today. The Menti is at 7 p.m. So please do join. And the topic is biodiversity and conservation. Fine. So the anther, so now we are done talking about the filament, now talking about the anther. The anther is terminal and it is bilobed. So this is how I have drawn the anther, right? So it is at the terminal portion of your stamen and it is bilobed here. So each lobe has two theca, meaning it has two divisions. So it is dithecous and it is a longitudinal groove that runs lengthwise. So if this is my anther which is bilobed, so I have a theca like this. So I have a division like this. So this is the structure of my anther. Okay. Now inside this I have a theca, meaning a division or a wall or a septum. Okay. So it means it is dithecous. Fine. So point number one, we are talking about your stamen. Your stamen has two structures, a long slender filament and a terminally bilobed anther. Okay. So anther has a cornered structure and it has 4 microsporangia in each of the 2 lobes. Okay. So in each of the, so it has 2 lobes, so it is terminally bilobed, and in each of the 2 lobes it has 2 microsporangia. Your microsporangia develops into pollen sac and your pollen sac produces your pollen grains. Alright, your pollen sac will produce your pollen grains. So this is how it is. So do I have a picture? Yes. So this is your stamen. Okay. So here you have a filament. Here is your anther. So the anther is terminal and it is bilobed, and it also has a theca that runs longitudinally lengthwise. So this is your theca. So that is why it is dithecous. Okay. So this anther is a cornered structure. So it has 2 microsporangia in each of the lobe. So this lobe has 2 microsporangia. This lobe has 2 microsporangia. Alright. So this is along the breadthwise. So each one develops into a potential pollen sac. Sac means a bag, and each one produces pollen grains. Your pollen grains are your male gametes. Yes ma'am, good evening. So thank you so much for joining my session. So please do like, share and subscribe. Alright, is this clear? So you understood the structure of stamen. So what is anther, what is filament, and how the anther is dithecous, and what is microsporangia. So microsporangia develops into pollen sacs, and pollen sacs produces your pollen grain. Okay, so shall I move forward? Okay, so structure of a microsporangium. The structure of the microsporangium, which is your pollen sac, which is going to develop into, which is your pollen sac. Microsporangia develops into microsporangium, that is the pollen sac. So it has 4 layers. The wall of microsporangium has 4 layers. The first layer is epidermis, the second one is endothecium, the third one is your middle layer, and the fourth one is your tapetum. Anthony Abhi, are you making notes? So should I slow down, or is my speed fine? Should I slow down? Are you able to make notes here? Okay, fine. So you have 4 walls of your microsporangium. So it starts with your epidermis, endothecium, middle layer and your tapetum. Okay, the first 3 layers have a similar function. So they help in the protection of the anther and dehiscence of the anther so that it releases the pollen grains. So what is this dehiscence of anther? In these cases, mitosis and meiosis is confusing. Don't worry, I have still not taught you, right? So I will teach you so that you will get it cleared. Okay, fine. So don't worry, it is not confusing. It will be clear. Okay, yes. So the first 3 layers, epidermis, endodermis and middle layer, have the same function. So they protect the anther and they help in the dehiscence of anther so that they can release the pollen grains. So I told you inside the anther, so it is like this. So inside the anther you have pollen sacs like this, and pollen sacs have your pollen grains. Now your pollen grain has to come out, right? Only then your pollination, your fertilization can happen. So for that your anther wall has to open. So that is called as dehiscence. So the first 3 layers of your microsporangium, they provide protection to the anther as well as help in the dehiscence of anther so that your pollen grains come out. Alright, is this clear? Fine. So now the last layer, that is tapetum, it has a very important role to play. So tapetum nourishes the developing pollen grains. So just like your placenta provides food to the growing embryo, so here it is your tapetum that nourishes the developing pollen grains. Alright, so these are the 4 wall layers of your microsporangium. Clear? So now I move forward. So the cells of the tapetum, so this tapetum layer has cells that is going to nourish the developing pollen grains. But how are the cells of the tapetum? They have a dense cytoplasm and they have more than 1 nucleus. So why do they have more than 1 nucleus? It's because of a condition known as endomitosis. Because of a condition known as endomitosis. What is endomitosis? So in a cell division, only when the nucleus divides and the cytoplasm does not divide, so that is called as endomitosis, where karyokinesis is not followed by cytokinesis. So a cell ends up having more than 1 nucleus. So the cells of tapetum have more than 1 nucleus because of this endomitosis condition. Okay, yes Harini, good evening. So thank you so much for joining us. So please do like, share and subscribe. Alright. Okay, so this is about a tapetum. So make sure you remember the endomitosis process. Another point, so when the anther is very young, so there is a group of compactly arranged homogeneous cells which is called as sporogenous tissue. So in the center of your microsporangia, so in the center of your microsporangia, so this is your microsporangia, right? So in the center, when the anther is very young, it has a group of compactly packed homogeneous tissue, so that is called as your sporogenous tissue. Alright, so that is your sporogenous tissue. So now let's go forward. So this is present at the center of each microsporangium. So each of the pollen sacs, so you have 4 pollen sacs in a bilobed anther. So in the center, when the anther is young, you have your sporogenous tissue. So this is the structure. So this is your microsporangium and it has 4 layers. What are the layers? It is your epidermis, endothecium. Then you have a middle layer and finally you have your tapetum. So in the middle of each microsporangia, so this is your microsporangia, right? So this entire thing is your microsporangium. So microsporangium contains your microsporangia. So each one is a microsporangia, alright? So microsporangia develops into your pollen sac, fine? So the outer thing is called as microsporangium. The inner 4 things are called as microsporangia, which is your pollen sac, and inside that is where your pollen grains are going to be produced. So at the center of each microsporangia, you have a sporogenous tissue. Alright, is this much clear now? So no doubt. So cells of tapetum have more than 1 nucleus because of the process of endomitosis. Okay, so now shall I move forward? Yes? Okay. So now what is microsporogenesis? So always remember genesis. So okay, so G is missing. So genesis means formation. So genesis is a Greek term which means formation. So microsporogenesis. So if you remember plant kingdom, so we spoke about microspores and megaspores, right? So microspores means micro is small, mega is big. So formation of minute spores. So minute spores here are your male gametes. So we call your male gametes in case of angiosperms as microspores. The formation of microspores is called as microsporogenesis. Alright? Okay. So now I move forward. So anther develops. So as the anther develops, so in the center of the anther, microsporangium is covering of microsporangia. Yes, you're perfectly right. So sporangium is the bag inside which you have microsporangia. Microsporangia develops into pollen sac which produces your pollen grains. Alright, is this much clear? No doubt? Fine. Now, in the center of your microsporangia, you have your sporogenous tissue. So when is the sporogenous tissue present? When your anther is young. But when the anther starts developing, so this sporogenous tissue, it undergoes meiotic division to produce your microspore tetrads. So, in the center of your microsporangia, you have a group of compactly packed cells which are called as sporogenous tissue. So, this is when the anther is young. So, when this anther begins to grow, your sporogenous tissue, it undergoes meiotic division to produce microspore tetrads. So, tetrads means 4. It produces 4 microspore cells. Okay. So, thus each sporogenous tissue is a potential pollen or microspore mother cell. So, it is the mother cell which is undergoing meiotic division to give your microspore tetrads. Alright. So, this pollen mother cell which undergoes meiosis to produce your microspores is called as microsporogenesis. So, the process by which your pollen mother cell present inside your sporogenous tissue, it undergoes meiosis to form microspores, that is called as microsporogenesis. Okay. Now, microspores are always arranged in a cluster of 4. So, that is why it is called as microspore tetrad. Alright. So, when the anther mature and dehydrate, so the microspores which are always present together as clusters will dissociate from each other. Then they will develop into a pollen grain. Alright. Okay. So microsporangium, that is the pollen sac, that is that entire thing, will have thousands of microspores or pollen grains, and they will be released by the process of dehiscence of anther. So once again, let me make it clear. So this is your microsporangium. Okay, so it has 4 layers. Then it has 4 microsporangia. So these are microsporangia. So now I am drawing 1 microsporangia here. So in the center I have a group of tissues. So this is my sporogenous tissues. So what is this? This is my microsporangia. So this is microsporangia. What is this? This is microsporangium. Fine. So this is when the anther is young. But when the anther begins to grow, your sporogenous tissue contains cells called as pollen mother cells or gamete or spore mother cells. Pollen mother cells or spore mother cells. Then this undergoes meiotic division to produce 4 microspore tetrads. So this process of sporogenous tissue containing your pollen mother cell undergoing meiotic division to produce your microspore is called as microsporogenesis. Clear? Yes? Okay. So always it is produced in a cluster of 4 cells, so that is why it is called as microspore tetrads. But as the anther matures and dehydrates, so each of them dissociate and they become an individual pollen grain. Fine? So this is the process of microsporogenesis. Any doubt? Yes? Till this much it's clear? So shall I move forward? So now what is the ploidy of your microspore mother cell or your sporogenous tissue? So your sporogenous tissue consists of your pollen mother cell. So what is the ploidy of the pollen mother cell? Haploid. So here you have 4 microspore tetrads and each one will develop into a pollen grain. So each of the tetrad will be haploid, because at the end of meiosis you get 4 haploid cells, right? So that is how it happens. So how meiosis happens? So you have a mother cell who is diploid. So mother cell produces 2 haploids. So this is meiotic division 1. So this in turn will produce 2 haploid cells and this will produce 2 haploid cells. So this is meiotic division 2. So here you have 4 haploid cells. The same thing is happening. Pollen mother cell undergoes meiotic division to produce 4 microspore tetrads. So each one will be a haploid cell. Yes, Pooja Kumari, hello, hi. So thank you so much for joining my session. So please do like, share and subscribe. So today I have a Menti quiz at 7 p.m. So please do join me, and the topic is biodiversity and conservation. So is this clear? So you understood what is microsporangium, microsporangia, and what is a sporogenous tissue, what is microsporogenesis, what is a pollen mother cell, and what is a microspore tetrad? No doubt. So shall I move forward to the next part? Alright. So now we are going to talk about the pollen grain. So the pollen grain represents the male gametophyte. So pollen grain is a small spherical structure like this. Okay, so it has a diameter of about 25 to 50 micrometer. So it has prominent 2-layered wall. So your pollen grain has a 2-layered wall. Okay, so the outer layer is called as XII and the inner layer is called as intine. So remember, the external layer is XII and the internal layer is your intine. So E and E is the external layer, I and I, it is intine. So exine and intine are the 2 layers of your pollen grains. So this is how it looks. So this is your pollen grain. It has an inner layer. This is your intine. It has an outer layer, which is your exine. Alright, the outer layer, so we'll talk about each of the layer. So first talking about the outer layer, that is your exine. Alright, so it has a hard outer layer. So the outer layer is very hard. So the outer layer is exine and it is very hard and it is made up of a particular type of substance which is called as sporopollenin. So the exine is a very hard layer because it has a substance, or it is made up of a substance called as sporopollenin. So this is the most resistant organic submaterial known till date. Okay, so nothing can resist, nothing can break it down, nothing can destruct it. All right, so this sporopollenin can withstand very high temperature. At the same time, it can withstand very strong acids and alkalis, but still it remains very intact. So that is your sporopollenin. So, the pollen grain exine has a prominent aperture, has a prominent structure called as germ pores, where your sporopollenin is absent. So, once again going to my previous slide. So, can you see the outer layer exine? Yes. So, here you have an outer layer that is exine, and on the outer layer you have certain apertures, certain places which are called as germ pores. Can you see? Here is a germ pore, here is a germ pore, here is a germ pore. So this germ pore is one point on the exine where your sporopollenin is absent. So this entire outer layer is made up of sporopollenin except at germ pore layers where your sporopollenin is absent. Alright, is this much clear? Okay, so then moving forward, that is about your sporopollenin. So pollen grains are well preserved as fossils because of your sporopollenin. Because your sporopollenin is very resistant to any type of treatment. So it protects the pollen grain very properly. So thus you can use it as a fossil as well. So recently they found a plant from your Arctic tundra region. So it was a seed and it was able to germinate. So sometimes your pollen grains and your seeds, they never fail to fascinate us. So this can store the pollen grains so very well from all the external conditions. Okay, so that is why it is preserved as fossils. Fine. Now talking about the internal layer, that is your intine. So your intine has an inner wall. So it is a thin continuous wall. The outer one is a thick wall. The inner one is a thin continuous wall. So it is made up of cellulose and pectin. So the outer layer is made up of sporopollenin, the inner layer is made up of cellulose and pectin. Alright, so the cytoplasm of the pollen grain is surrounded by your plasma membrane. So the pollen grain also has a plasma membrane as well as a cytoplasm. Alright, yes. So did you understand what is an exine, what is an intine? So no doubts, things are clear? Yes, so shall I move forward to the next part of the pollen grain? Yes? Okay, so when the pollen grain matures, so your microspore tetrads will develop into pollen grain. So when the pollen grain matures, it will have two cells. Okay, so what are the two cells it will have? So one is your vegetative cell, alright, the other one is your generative cell. So these are the two cells. So when the pollen grain matures, it will have two cells. One is vegetative cell, the other one is your generative cell. So vegetative cell is the bigger of the two cells. Okay, so it has two cells. Out of these two, vegetative cell is bigger and it is abundant. It has abundant food reserve. It is large. It has a large irregularly shaped nucleus as well. Whereas the generative cell is small and it floats in the cytoplasm of the vegetative cell. So you have a big cell, that is vegetative cell, and this has a large irregularly shaped nucleus. So within the cytoplasm of your vegetative cell, you have a small cell that is called as your generative cell. Fine. So this spindle shape, so this generative cell is spindle shaped with dense cytoplasm and a nucleus. So your generative cell also has a nucleus. When the pollen grain is shed at this stage, meaning when the pollen grain comes out of the anther at this two-cell stage, this is called as two-cell stage release of pollen grains. So when the pollen grain comes out of the anther, containing two cells, that is vegetative cell and generative cell. It is called as the two-cell stage of the pollen grain. Alright, is this clear? What is the two-cell stage of the pollen? Fine. So you have your vegetative cell, so it is a very big cell, okay. So it has an irregularly shaped nucleus. So inside the cytoplasm of the vegetative cell, you have another cell, that is your generative cell. So this generative cell is spindle shaped. It also has a dense cytoplasm and a nucleus. Alright. So when the pollen grain comes out of the anther at this stage, it is called as the two-cell stage. Clear? Yes? So shall I move forward? So now did you understand the outer layer and the inner layer of the pollen grain? What is XII, what is intine, and where these two cells come from? Yes, so as the pollen grain matures, it will have two cells. One is vegetative, one is generative. So vegetative is a bigger one, generative is a smaller one. Fine. So shall I move forward? Yes? Okay. So 60% of the angiosperms, they shed their pollen grain at this particular stage. At what stage? At what stage? At the two-cell stage, alright. So the remaining angiosperms, what happens? They do not shed the pollen grain at the two-cell stage, but instead there the generative cell divides mitotically to form two male gametes before it is shed. So in majority of the angiosperms, okay, in almost 60% of the angiosperms, your pollen grains are shed at this two-cell stage, containing a vegetative cell and a generative cell. Alright, but in the remaining angiosperms, what happens is that the small generative cell, it undergoes mitotic division to produce two male gametes. So which means your generative cell has a haploid nucleus. So when it undergoes mitosis, it will produce two haploid male gametes, and when the pollen grain is shed at this stage, it is your three-celled stage. So now you understand what is two-cell and what is three-cell, where the three-cell comes from. So three-cell is when your generative cell divides mitotically to form two male gametes. So once again, here we go. So you have your microspore mother cell, or it is known as the pollen mother cell. So where is this pollen mother cell present? In your sporogenous tissue, right? So where is your sporogenous tissue present? In your microsporangia, which is inside your microsporangium. Now this pollen mother cell will undergo meiosis. So at the end of meiosis, you have four haploid cells. This is your tetrad of microspores, or this is called as four microspores. Now each of the pollen grain will undergo mitosis to produce a vegetative cell and a generative cell. If the pollen grain is shed at this stage, it is called as the two-celled stage. Okay, when this generative cell alone further undergoes mitosis, it produces two male gametes. So that is your three-cell stage of the pollen grain. Alright, yes. Now did you understand this process, how it happens? Okay, hello Rushi, hi, so good evening. Thank you so much for joining our session. Yes, you are here after a very long time. So how are you doing? So done with all your school exams? So now you're ready to listen to class 11 portion? Yes? Alright, fine. So anyway, thank you so much Rushi for joining us. So please do like, share and subscribe. So today I have a mentee, so please do join. I know it is a class 12 topic. So this syllabus for mentees, biodiversity and conservation, but still give it a try. It is a general topic. Alright, so now I hope you understood how the pollen mother cell gives you a microspore tetrads, how you get vegetative and generative cell, and how you get three-cell stage of a pollen. Yes? For all those who are watching, is this clear? Should I explain once again? Yes, let me know quick. So you should never make a mistake. So look at the ploidy of each of them. Yes, is this clear? So this vegetative cell has N and your generative cell has a ploidy of N. So again the male gametes will be N and N. Okay, so you should know the number of chromosomes, whether it's haploid or diploid in each case. Fine? Alright, okay. So now I move forward to the next part. Okay, So pollen grains, so many species, so nowadays pollen grains serve as allergy for allergies, and it can also cause bronchial affiliations which can lead to respiratory disorders like your asthma and bronchitis. Okay, so that is why it is always recommended don't go for a walk in the early morning, because early morning is where your pollination is at its peak. Okay, if at all you encounter a pollen in your way, you can become allergic to your pollen. Okay, so this pollen allergy is very common. Alright, so this Parthenium grass of carrot was an imported pollen grain along with, it was a pollen, it caused a lot of pollen allergy and it came to picture with your imported wheat. Alright, yes? Fine. So pollen grains are rich in nutrients and it can also be used as your supplements. So in the form of syrups and tablets, it can be used because your pollen grains are rich in nutrients. And who supplies the developing pollen grain? It is your tapetal cells. Alright. So it is actually very rich in a lot of nutrients and it can be used as supplements in the form of tablets and syrups. So it is usually consumed by your athletes and race horses to increase their performance. Fine. So pollen grains on the stigma. So now your pollen grains are ready. Your pollen grains are produced. Now your pollen grain has to land on the stigma before they lose their viability, right? Yes. So this pollen grain, so before it reaches the stigma, it has to be pollinated. But it has to be pollinated within a period of time so that it does not lose its function as such. Okay, so viability is the life of the pollen grain. It should not become inactive by the time it reaches the stigma. Alright, so that is called as the viability. So, but how long is the viability of the pollen grain? For how long can this pollen grain stay functional? So, this viability is highly variable and it depends upon the prevailing temperature and humidity. So, these are the two conditions on which the viability of a pollen grain depends. So, here you have a few examples. In case of cereals like your rice and wheat, pollen grains can lose viability within 30 minutes of their release. So, release from where? From the anther. Okay, they can either be shed at the 2-cell stage or the 3-cell stage, but within 30 minutes in case of cereals like your rice and wheat, they have to be transported to the stigma. Otherwise, they will become non-functional. Okay. So, in case of plants like your Rosaceae, Leguminaceae and Solanaceae, they maintain viability for many years. Alright, so this viability is highly variable in a lot of cases. You can also store pollen grains in liquid nitrogen. So, liquid nitrogen is in -196 degrees. So, it is a type of cryopreservation. This is your liquid nitrogen. So, you add your cryopreservatives to your pollen grain and store it in this nitrogen containing tank and put this in -196 degrees Celsius so that it becomes dormant. It enters a dormant stage, but it can be got back to viability. You are arresting the functions. You are making the pollen enter a dormant stage. So, by putting it in -196 degrees Celsius. Alright, it is similar to your seed bank. So, this can be called as a pollen bank. So, there are ways of preserving your pollen grains. Alright, is this clear? So, with this we are done with the first topic, that is your stamens, megasporangium and your pollen grains. So, you understood everything from this? Yes? Okay, hello, hi. Yes, I remember. So, Nirmai, hello. So, you told me your 10th standard is over, right? So, you are joining as my student. So, now you are ready to listen to class 11 topics. Yes, but anyway, thank you so much for joining me. So, please do like, share and subscribe. Nirmai, alright. So, now is the first part of this chapter clear? Yes? So, no doubts? Okay, so this is my last slide. So, I don't have a slide after this. So, this is a gentle reminder. So, I have a Menti quiz in the topic biodiversity and conservation and the time is 7 p.m. this evening. So, make sure you are present. So, we are not going to play for very long. So, it is usually a 25 minutes Menti. So, 15 questions, 20 seconds each. So, make sure you come. Alright. So, the first part of the chapter is done. In the next class, I will talk about the pistil, megasporangium and the formation of your ovary. Formation of your egg, that is your female gamete. Here it is called as an embryo sac. Alright. So, do you have any other doubts? Yes? So tomorrow, yes, another important announcement. So tomorrow 4 to 5, we are talking about your central nervous system, because yesterday we started off with your human nervous system. So I am done explaining the structure of your neurons yesterday. So tomorrow we'll talk about the transmission of impulse between your neurons and the structure of your human brain. Okay? Yes? And tomorrow at 7 o'clock, I have another session with you where I am starting your biomolecules chapter. So tomorrow you're going to have 2 live sessions, 4 to 5 and 7 to 8. 4 to 5 we'll be talking about the structure of your brain, and 7 to 8 I'll be starting your biomolecules. So biomolecules will be from 7 to 8. So this session from 7 to 8 will be continuous on Monday, Tuesday, Wednesday. So where we will complete your biomolecules. Alright? Yes? So make sure you are present for both the sessions tomorrow. So before that, you meet me today at 7 o'clock where we'll be having a mentee. So you have started your preparation of NEET. Thank you so much, Nirmai. So if you do not understand, so please stop me. I will slow down and explain again. Okay? So guys, start planning. So don't waste time, your NEET exam. So as the days are passing by, so the time that you have for NEET exam is reducing. So make a proper plan, make a proper strategy. So how much you want to score, what is your target, so which college you have to land up. So please have a lot of motivation so that you finish your portions beforehand. So don't extend your portions till the last minute. So you should at least do a minimum revision of 2 rounds, but I would suggest for a perfection, so it should be 4 revisions. Alright, so before you do revision, you should first finish studying, right? So make up a proper plan. If you have any doubts, so get back to me in biology so I can help you work out the plan. So if you want to talk to me, so there is a Telegram link beneath my description box, beneath the video. So get there. Okay, so there we will talk about all your doubts so I can help you out with biology. You want to get into CMC Vellore, alright, so that is one of the best premium colleges. So all the very best. So all my wishes for you. I want to see all of you with flying colors. Alright, so I want to see all of you as doctors very soon. So for that, yes, all my blessings and wishes to you. So very soon I want to see you as doctors. For that, I want all of you to clear NEET exam. So NEET exam is one small test for your entire journey. So just clear this test with a lot of desire and admiration. Okay, so don't take it as a frustration. Okay, there is someone new who's called as Sanjay. Hello, hi. So thank you so much for joining. So I guess you're new to my channel. So please do like, share and subscribe. But I'm so sorry, I'm done with my session and you're just here. So I take class from 4 to 5. So try coming at 4 o'clock. Otherwise, watch my recorded lecture. So 6:30 I have my mentee in 7 o'clock I have my mentee in biodiversity and conservation. So please do join me. And tomorrow we have 2 classes, 4 to 5 we're going to talk about the structure of the brain, and 7 to 8 we're going to start talking about biomolecules. Alright? Okay, chalo. So thank you for being such a wonderful audience. So I'm waiting to meet you at 7 p.m. So please prepare, revise and come for my mentee. So until then, stay tuned, take care and bye-bye.